Welcome to biology, the scientific study of life. Species diversity. The species diversity of a community, the variety of different kinds of organisms that make up the community has two components. One is species richness, the total number of different species in the community. The other is the relative abundance of the different species. The proportion each species represents of the total individuals in the community. For example, imagine two small forest community, each with 100 individuals distributed among four different tree species A, B, C. Notice that as shown here. A, B, C, D. As follows: Community A 25%, B 25%, C 25%, D 25%. Community 2, picture 2, community 2, A 80%, B 5%, C 5%, D 10%. Notice that. The species richness is the same for both communities because the both contain four species, but the relative abundance is very different. You would easily notice the four different types of trees in community 1, but without looking carefully, you might see only the abundant species A in the second forest. Most observers would intuitively describe community 1 as the more diverse of the two communities. Indeed, for an ecologist, the species diversity of the community is dependent on both species richness and relative abundance. Determining the number of species in a community is easier said than done. Various sampling techniques can be employed, but since most species in a community are relatively rare, it may be hard to come up with a sample size large enough to be representative. It is particularly difficult to accurately census the highly motile or less visible members of communities, such as mites and nematodes. Nevertheless, measuring species diversity is essential not only for understanding community structure but for conserving biodiversity. Trophic structure. The structure and dynamics of community depend to a large extent on the feeding relationships between organisms. The trophic structure of the community, the transfer of food energy up the trophic levels from its source in plants and other photosynthetic organisms, primary producers through herbivores, primary consumers to carnivores, secondary and tertiary consumers as shown in this figure, and eventually to decomposers is referred to as a food chain. Notice that this is a marine food chain and a terrestrial food chain. Plant primary producers, herbivore primary consumers, and carnivore secondary consumers, and tertiary consumers, carnivore quaternary consumers. Food webs. In the 1920, Oxford University biologist Charles Elton recognized that food chains are not isolated units but are linked together in food webs. An ecologist can summarize the trophic relationships of a community by diagramming a food web with arrows linking species according to who eats whom. For example, is a simplified food web for an Antarctic pelagic community. The primary producers in the community are phytoplankton, which serve as food for the dominant grazing zooplankton, especially euphausiids, krill, and copods, both of which are crustaceans. These zooplankton species are in turn eaten by various carnivores, including other plankton. penguins, seals, fishes, and baleen whales, squids, which are carnivores that feed on fishes as well as zooplankton, are another important link in these food webs, as they are in turn eaten by seals and toothed whales during the time when whales were commonly hunted for food. Um, Notice that also, as shown this figure, humans became the top predator in this food web, having hunted many whale species to low numbers. Humans are now harvesting at lower trophic levels, catching krill as well as fishes. Who are food or how are food chains linked into food webs? First, a given species may weave into the web at more than one trophic level. For example, in the Antarctic food web, euphosides feed on phytoplankton as well as on other grazing zooplankton, such as copepeds. Such non-exclusive consumers are also found in terrestrial communities. For example, foxes are omnivores with diets that include berries and other plant material. Herbivores such as mice and other predators such as weasels. Humans are among the most versatile of omnivores. Food webs can be very complicated, but we can simplify them for easier study in two ways. First, we can group species with similar trophic relationships in a given community into broad functional groups. For example, as shown in this figure, more than 100 phytoplankton species are grouped as the primary producers in the food web. A second way to simplify a food web for closer study is to isolate a portion of web that interacts very little with the rest of the community. As shown this figure, notice that. This figure illustrates a partial food web for sea nettles, jellies, and juvenile striped bass in Chesapeake Bay. Notice that. Limits on food chain length. Each food chain within a food web is usually only a few lengths long. In the Antarctic web, as shown this figure, there are rarely more than 7 links from the producers to any top level predator, and there are even fewer links in most chains. In fact, most food webs studied in date consist of 5 or fewer links, as Cheryl Elton first pointed out in the 1920s. Why are food chains relatively short? There are two main hypotheses. One is the energetic hypothesis, suggests that the length of food chain is limited by the inefficiency of energy transfer along the chain. Only about 10% of the energy stored in the organic matter of each trophic level is converted to organic matter at the next trophic level. Thus, a producer level consisting of 100 kg of plant material can support about 10 kg of herbivore biomass and 1 kg of carnivore biomass. The energetic hypothesis predicts that food chains should be relatively longer in habitats of higher photosynthetic productivity, since the starting amount of energy is greater. A second hypothesis is dynamic stability hypothesis, proposes that long food chains are less stable than short chains. Population fluctuations at lower trophic levels are magnified at higher levels, potentially causing the local extinction of top predators. In a variable environment, top predators must be able to recover from environment shocks such as extreme winters. that can reduce the food supply all the way up the food chain. The longer the food chain, the slower the recovery from environmental setbacks for top predators. This hypothesis predicts that food chains should be shorter in up or in unpredictable environments. Most of the data available support the energetic hypothesis. For example, ecologists have used tree whole communities in tropical forests as experimental models to test the energetic hypothesis. Many trees have small branch scales that rot, forming small holes in the tree trunk. The tree holes hold water and provide a habitat for tiny communities consisting of decomposers or decomposer microorganisms and insects that feed on leaf litter, as well as predator insects, as shown in this figure. Shows the results. This figure shows the results of a set of experiments in which researchers manipulated productivity leaf litter falling into the tree holes as predicted by the energetic hypothesis. Holes with the most leaf litter and hence the greatest total food supply at the producer level supported the longest food chains. Another factor that may limit food chain length is that animals in a food chain tend to be larger at successive trophic levels, except for parasites. The size of an animal and its feeding mechanism but some upper limit on the size of food it can take into its mouth. And except in a few cases, large carnivores cannot live on very small food items because they cannot procure enough food in a given time to meet their metabolic needs. Among the exceptions, there are baleen whales, huge suspension feeders with adaptations that enable them to consume enormous quantities of krill and other small organisms. Back to test of this figure represents or shows the results. Test of the energetic hypothesis for the restriction of food chain length. Researchers manipulated the productivity of tree hole communities in Queensland, Australia by providing leaf litter input at 3 levels: high litter input, natural, control, rate of litter fall medium equal 1 distribution 10 natural rate, and low 1 distribution 100 natural rate. Reducing energy input reduced food chain length, a result consistent with the energetic hypothesis. Notice that.